Hello, everyone. Welcome to our early morning Sunday thing. It was really great you can come today. We're very grateful. Today, it's quite an action-packed one. There's a lot of biochemistry, which is quite diverse, and you're going to start your ethics, which will inform your assignment later on. So we've got quite a lot to go through, so we'll get started.
So before we go into the content, we've introduced a bit of a yield system to help you guys prioritize your learning. Because there is a lot to get through, certain topics are more important than others. So sometimes you just need to understand the big picture. So don't need to know all the details, just the main ideas. Other times, topics could be very high yield, so we definitely recommend you guys focus on those if you're short on time. So throughout, we've sort of like labeled each slide, and yeah, it'll give you a better idea of it.
So genomic medicine first up. So an important factor of understanding genomic medicine is understanding the types of different genetic variations and gene defects. So there's two important concepts for you to understand in a big picture sense. The first is monogenic disorders. Monogenic disorders are typically rare and they involve a single defective gene. Examples of this include cystic fibrosis and hyperlipidemia. They're often very easily predicted and highly associated to the disease. There's not many environmental factors necessarily. Whereas multifactorial, for example, type 1 diabetes, are when a combination of many different inherited gene variations, as well as different environmental stimulus and factors, contribute to the development of a condition. So monogenic, single defective gene leads to disorder, multifactorial, many variables. Next slide, please. So what is a mutation and what is an SNP, or a single nucleotide polymorphism? On average, there is a difference of about 1 in 1,000 base pair difference between people. And if you consider that the human genome has about 3 billion base pairs, there's about 3 million base pair variations between two people. So which of these are mutations and which of these are single nucleotide polymorphisms? A single nucleotide polymorphism is different to a mutation because there's a DNA variation in which a single base pair varies among people of a species, but must occur in at least 1% of the population to be an SNP rather than a mutation. SNPs are can be targeted in a more clinically relevant scenario because you can make suitable medication, as we'll see later, for those typical people. SNPs can occur in exons, introns, and intergenic sequences. Intergenic sequences are also known as junk DNA, and they have unknown functions. 50% of SNPs occur in non-coding regions, which means they may impact promoter complexes and stability of transcribed strands. However, they're not necessarily going to have huge effects. Unlike the 25%, which are missense mutations, a missense is a point mutation that changes a single nucleotide. Sorry for butchering my French. And these alter the amino acid sequence of the protein, meaning the conformation of the protein may be altered, reducing its efficacy, stability, and binding and overall function. So these are serious and have physiological and noticeable effects in the body. 25% of SNPs are also silent, which means they have no noticeable effect. Okay, next slide. SNP and medication. Genetic variation greatly influences how drugs interact in the body, and pharmacodynamics, which is how they affect the body. SNPs can affect metabolite enzymes, receptor targets, and other anatomical structures important in the distribution and absorption of medication. There's a new type of medicine that is becoming popular known as personalized medicine, which considers individual gene variability and may allow disease susceptibility to be better understood by analyzing SNPs. For example, if you take a lot of type 1 diabetics and see what SNPs they all have in common, you can understand the disease greater. So there's things called genome-wide association studies, which correlate particular SNPs with certain diseases and may also show the defective proteins or genes that are responsible for the certain conditions that make people susceptible. It also can show you which drugs are more effective, because some drugs will interact in different ways depending on the function of the receptor. So you have a dysfunction in receptor, then you know not to give them too little of a drug, because the drug won't be metabolized. Whereas if they have a mutation that makes the drug not metabolized at all, so it's very saturated in the body, that's really important to know, and so you should give people less of the drug. And examples of these are warfarin and beta blockers. This isn't important, it was in the lectures, but it will be in your lectures. You don't have to understand it, but all it's saying is that the way that these two drugs are absorbed and distributed and metabolized depends on particular gene variations that affect the protein conformation. So I want to make sure, I'm just going to go over that again, because it's good to understand. If you have a mutation in this receptor, it may mean that that drug is not metabolized very quickly, which means there's a large concentration of it in the body, which means you shouldn't give those people too much drugs or they will overdose, for example. So it's important to understand how SNPs interact with drugs and contribute to disease for the clinical treatment of them. That's why this is important. Next one. And there's not much more than that.
Okay, so I'll be taking population genetics. With population genetics, they sort of like mention in the lecture, but then they don't bring it up too much again. You might have like one question perhaps in exam, so it's not too important. So just and it's kind of mathy, this particular section. So if you like maths, this might be fairly intuitive for you. So just to start off with, there's some terms that you should know. So you probably know all of these from year 12 bio or the bio bridging course if you did it. I know not all people do it, fair enough. So yeah, some basic terminology here. So trait, characteristic of an organism, something that's determined by their genetics and environment. So something like eye color. The phenotype is the actual manifestation of that trait. So it might be green eyes or brown eyes. A gene is basically just like a quantity of DNA that codes for something. The locus is where that gene is located on a specific chromosome. Allele is a specific version or alternate version of that gene. Genotype refers to the genetic composition of the individual for a trait. So you might know you have two chromosomes, like two autosomal chromosomes. So you might have two chromosome 1s, two chromosome 2s, all the way up to 23. And so you have a version of the gene on each chromosome. So that might be big A, little a. Big A refers to dominant, little a is recessive. So yeah, those are your different combinations. And if you're homozygote, that means you have the same alleles on each of the chromosomes at the locus. So that's AA, both capital A or both little a. And then you have heterozygote, which means the two chromosomes have different alleles on each of the two chromosomes. So you might have big A on one chromosome and a little a on the other chromosome. And gene pool just refers to all copies of all the genes in a population. And yeah, so you can see it kind of gets a little mathy here. I can't sit in chat at the moment, so if there are any questions that are relevant, I hope one of the PSP people can just like shout at me. So say that capital A or big A is a gene for brown eyes and little a is a gene for blue eyes, and pretend we have a population of 64, which isn't that big, but it's an example. So let's say 12 people have little a, 38 people have big A, little a, and 14 people have big A, big A. So the phenotype is the physical manifestation of the trait, right? So the people with little a will have blue eyes, the 38 and the 14 people, because they have a big A, they're dominant, and they all have brown eyes. So if you just calculate the frequency, it's just a proportion of the people with a particular phenotype. It's quite simple and straightforward. So the proportion of people that have blue eyes will be 12 over 64, which is 19%. And the people that have brown eyes will be 52 over 64, which is 81% for brown eyes. And you can also have allele frequency, which don't get scared by the equation too much. It will make sense, sort of. It'll sort of make sense, hopefully. But anyways, allele frequency just refers to the proportion of an allele in the population against like the total number of alleles. So the proportion of or frequency of the dominant allele, which is big A in this case, we represent using P. And for the recessive allele, we use Q. So if you look at the equations on our right, P is for our dominant allele, and because and this N big A here is just referring to the number of people who have double A as their, or double big A as their genotype. So that's 14 people here, and it's plus the number of the people who have one big A and one little a, which is 38. So you're basically finding the number of big A's in the population. So this is why you have the times 2, because the people with 2 big A's, right, they have 2 big A's. So you'll need a times 2 for, so each of the 14 people, you'll have to times by 2 because they have 2 big A's. Hope that makes sense. And for the people who are heterozygote, which means they only have one big A and one little a, you don't need to times 2 because they only have one big A in there. So basically the top is how many big A's there are in the population. And then you divide it by 2n. 2n just refers to the total number in the population. So that's 64. You need to times 2 because each of them have 2 genes, right? So the total number of genes will be 2 times the amount of people. So if we sub everything in, we have 14 people. We have 14 people here with 2 capital A, 38 people with 1 capital A. We need to times the people with 2 capital A's by 2 because they have 2 A's in their genotype. So the top gives us the total of how many big A's there are, and then the bottom is how many alleles there are in total. So 64 people times 2, because each individual of the 64 people have 2 alleles. So if you do the math, that works out to be 52%. And you apply the same idea on the bottom, but instead of instead of doubling the big A's, you double the number of people with little A's. And that's basically the same idea. Again, you don't times anything for the people who are heterozygote because they only have one little a. So the top finds the number of little a's in the whole population, and the bottom is basically the number of alleles again. And that works out to be 48% here. Yeah, so I hope that makes sense. If it doesn't, just let me know and I'm happy to go through it again, but I can't see the chat, so one of the PSP members, please keep an eye out. Okay, and then there's something called the Hardy-Weinberg equilibrium. So this equilibrium has a law, or basically states that allele frequencies, which is what we just looked at, do not change over generations, assuming that it is a large randomly mating population and that there is an absence of mutation, migration, or selection. So basically anything that can affect the frequencies. So because there's such a strong requirement to fit this kind of law, it has to be large randomly mating, it has to not have mutations, migrations, or selection. It's a pretty hypothetical sort of law because you'll basically never see it in nature that meets all of these conditions, but it's still useful for predicting genotype frequencies from allele frequencies through the equation below. So if you remember from the last slide, this P here is the frequency of the dominant allele, and this Q here is the frequency of the little allele. So because the P is the frequency of the dominant allele, right, then the proportion of the population, or the percentage of the population with a genotype of AA will be P times P, which is P squared. So you're kind of like saying, oh, the chance of, because this is P is the chance of getting a dominant allele, then getting 2 of them is basically P times P, or P squared. So if you apply the same reasoning, the genotype frequency of big A, little a is 2pq, which is really just P times Q, but you times it by 2 because it can either be little a, big A, or it can be big A, and then little a. And you have AA, which is just Q squared. So what this equation is really saying is that the number of big A, I really wish I could draw faster, but I'm using a trackpad, so I'm sorry about that. So what this equation is really saying is the proportion of people with big A, which is P squared, plus the proportion of people with big A, plus the proportion of people with little a, or equal to 1, or 100%. And that makes sense, right? Because if you, that's all the combinations you can get: big A, little a, and little a. That's all the combinations you can get, and if you add them all together, that should add up to the whole population, or 100%. So if you work out this allele frequencies for two generations of a population, and you see that they're basically the same or equal, then you can say the population is in the equilibrium. And there's some factors that disturb the equilibrium. So one of them is sources of new genetic variation and new alleles. So this is mutations. Mutations are relatively rare because, well, your DNA is relatively stable. Your DNA doesn't change that fast, so they're relatively rare. And there are also mechanisms that alter existing genetic variation. So there's migration, which just means that individuals within the population are moving to another population, they're coming and going. So because they're coming and going, that means their genes are sort of shifting between populations, and that can change the frequencies. You can have positive or negative selection, which is basically survival of the fittest. Positive is like, you're doing well, you're selected, you're positively selected and you'll survive. Negative means you'll die off. Genetic drift is just a chance fluctuation of the frequency of alleles generally in small populations, and non-random mating is what it sounds like. So in animals, you might see them inbreeding or outbreeding. So inbreeding, they look for other animals with the similar traits. Outbreeding, they look for other animals that have a bit different traits. So not too important, but these are some of the factors that you should know about. But the details of each one aren't too important. So here's an example you can use of the Hardy-Weinberg equilibrium. Let's say that the dominant allele is for black-furred cats, and it's represented by P. So 60% or 0.6, 60% of the total number of alleles is P, so the dominant allele for black-furred cats, and 40% of the alleles is for white-furred cats. So in addition to that, we know that the next generation of cats, there's 672 black cats and there's 128 white cats. And the question is the population equilibrium? So if you recall from what we said before, if we work out the allele frequencies, so P and Q, for both generations, and they're pretty much equal, then they should be in equilibrium. We say that it is in equilibrium. So we've been given that we've been giving the frequencies for the first generation. So P equals 0.6 and Q equals 0.4. With the second generation, we can figure out the allele frequencies. So if you notice, we have 128 white cats, right? So white is the recessive trait. So that means it's basically little a, or Q squared. That is supposed to be or. Drawing on the trackpad is very hard, struggles. So yeah, so the proportion of white cats in the population is Q squared because it's little a. So if the proportion is 128 over 800, that's 16% or 0.16. And so Q squared must equal 0.16 here. And if you do the math, then you square root it, it's 0.4. So if we compare the second generation, where Q equals 0.4, to what we had in the first generation, which is Q equals 0.4, that means the recessive allele frequency has remained the same. And because the recessive allele frequency has remained the same, then the P has to also remain the same, right? Because, well, P plus Q has to equal 1 because they make up all of the alleles in the population. There's only P and Qs. So the frequency of P's and the frequency of Q's has to add up to 1. And you can see that this does add up to 1, 0.4 plus 0.6 equals 1, or 100%, right? So because the recessive allele frequency and also the P frequency has remained the same, then the population is in equilibrium. Okay, I hope that makes sense. And here's another scenario or example that you can use with the equilibrium. I know it looks very mathy, but don't be too intimidated. It'll make sense. So here's a scenario. Say that a man has a sibling with a genetic disease that is rare, and he wants to know that if he has a child, what's the chance of his child also having the disease? So we know the condition is autosomal recessive, which means it's just not on a sex chromosome, and the gene is recessive, and that in general about 1 in 80,000 people are affected. So if 1 in 80, so if this is a recessive disease, that means they must have little a and little a to be affected and have the disease, right? So that's about 1 in 80,000 people. I'm just going to write 80K, otherwise it'll take me too long. And we know the number of people, or the proportion who have little a is equal to Q squared here, as we established in the previous slides. So that means the frequency of little a is 1 over 80,000, and that's equal to Q squared. So if we want to find just the proportion of the recessive allele, or Q, we can just take the square root of this. So if you square root it, then you find that Q equals 0.0035. And we know that P plus Q equals 1, as we said before, because the percentage of dominant alleles plus the percentage of recessive alleles has to equal 100%. It makes up all the alleles, right? So it has to equal 100%. So then we can find P. P will just be 1, or 100% take the proportion of Q, or yeah. So you get 0.9965 for P, and then the probability of being a carrier, which is big A, little a. So you're a carrier for the disease because you have a little a gene, but you don't actually have the disease because you have a big A gene that saves you, basically. That'll equal 2PQ, or 0.007, or 7 in 1,000 people. 7 in 1,000 people, right? Let me just clear this. And so moving on, we'll come back to what we worked out in the previous slide later, but just moving on, we know that the man's parents must have been carriers for the sibling to get the disease, but they couldn't have had the disease themselves because, well, they were disease-free, his parents were disease-free. So his parents must have been carriers, right? So they must have big A, little a, and for their genotypes. And I think you would have done Punnett squares in high school. So hopefully this looks familiar. So we know the parents, so we know this man, right? This man doesn't have the disease, but what's his chance of being a carrier? Because he doesn't have the disease, we can cross out that his genotype is not AA. So the man here, he could either be big A, or he could be big A, little a. So you see that there's 2 big A, little a's. So his chance of being a carrier is 2 out of 3, right? And so far, we know that the probability of the man's wife being a carrier from when we worked it out in the first slide is 7 out of 1,000. And then his probability of being carrier is 2 out of 3. if they're both carriers, then the probability of them both giving the child the recessive gene is half, right? So because, well, if they're both AA, then there's half chance they give the big A and there's half chance they give the little a. So the probability of the child getting the disease is 2/3. So that's the probability of the man being a carrier times 7 out of 1,000. That's the probability of the wife being a carrier times half. That's like half chance for the sperm to give the diseased allele, and then times half, that's half chance for the egg to have the diseased allele. And if you work that out, that's 0.001. So you can see that even though the man's brother or sibling, sister, had the disease, it's actually quite rare in this case for his child or offspring to have the disease, only 0.1%. So he's quite safe having a child. Oh, I did not mean to do that. Oh, sure, again, sorry about that. And now will be chromosomal disorders, and I think that will be David, I believe.
Yeah, Andrew, that was really, really great. That was really nicely done.
Thank you.
I know it's quite intimidating. There's a lot of numbers and concepts, especially that half times 2/3 thing is quite difficult. So we do put this up on YouTube. You can watch it if you get a chance. Take your time with it. It's not as intimidating as it looks. So right now we'll talk about some chromosomal disorders. Now I'm aware you haven't actually been through this, so we'll go nice and slow. Well, not too slow, but we'll tell me if you're confused and we'll go through it together. So chromosomes, as you will remember from your cell 1 and cell 2 lectures, are most visible and most condensed during the M phase of the cell cycle. At this phase, they're often used in karyotypes. Karyotypes are when they line genes up to see if there's any disorders or conditions. And they look at it when it's really condensed to clearly see the gene. So what will often happen is doctors will take some amniotic fluid from a mother. You don't have to know what that is yet, from a mother to see the karyotype of the baby, and they'll get nucleated cells during metaphase to look at the chromosomes. So all a chromosome is 2 chromatids. A chromatid is this little region over here. These are sister chromatids. Sister chromatids join together at the centromere, which is often visible. Okay. Chromosomes consist of chromatin, which is just DNA wound around protein complexes, such as histones. Histones are protein complexes that facilitate the packaging of DNA. And the reason why these stick together is because of things called proteins called cohesins. And during mitosis, these are broken apart, so sister chromatids are easier to separate. And just a fun fact, as the maternal age of the mother ages, there's less cohesin. It starts to break down and it's less reliable. So there's more reliable for there to be mutations for if an older mother is having a child. Next slide. This is just some nomenclature. This is mainly for notes. I'll explain it as we go along. So next slide. Again, the centromere determines the classification of the chromosome is a point of contact between these 2 chromosomes. Yep, you have 3 types that you really need to know, which are metacentric, in which the centromere is near the middle. So there's not really a notice. There's like the short arm and long arm are quite similar. There is a submetacentric where it's between the middle and end, which means there's quite a clear P for petite, short arm and long arm, Q. And acrocentric is when there's centromeres at one end. And these are called satellites, and they're often lost during mitosis or meiosis. So it's important to keep that in mind. Next slide. I'm not looking at the chat, so hopefully people in my thing answer, in the PSP answer. What is a karyotype? A karyotype looks like this. It is a collection of the pairs of homologous genes taken from a nucleated cell during the M phase of cell division. There are 22 pairs of autosomes, ranged from largest to smallest, with 1 pair of sex chromosomes, which are these little dudes. The Y chromosome is paternal, and a Y chromosome is only paternal. The mother's not going to give a Y chromosome. So again, I mentioned the amniotic fluid, and it's any nucleated cells that undergo cell division. Usually they take white cells because you can just take in a needle, take some blood and serum out, and you can have white blood cells. So it's quite easy. Next one. These are some high yield. This is quite examinable. They'll write in this notation, so it's important to understand. There are 46 chromosomes. The sex chromosomes are XY. This is a normal male. Down syndrome, there's an additional 21 chromosome. So you write plus 21. You know it's a female because, oh, you can't see my mouse. You know it's a female because it's XX. And this is almost always in exams, quite important. This stuff, the deletion and the inversions, are less high yield, and we'll explain them a bit later on. Next slide, please.
Yeah, I don't really think they'll give you deletion or inversion ones, but yeah, so just keep in mind at the top, you start off the number of chromosomes, number of sex, well, you write out the sex chromosomes and then you write the abnormality.
Yeah. And you could do minus 21 as well if that was a problem. This isn't important, but this is, just so you know, karyotype is when they take blood, nucleated white blood cells. It needs a nucleus, obviously, because that's where chromosomes are stored. And yeah, this isn't too important. Next slide. Band-in. So this is called G-band-in, and it assists in the identification of chromosomes, as well as revealing which genes are active, which are silent, and which genes are present. So if a band is very darkly stained, that means the DNA is very tightly wound. It's difficult for the transcription factors to go to their appropriate sites to facilitate transcription of the DNA into RNA. And so it is very condensed and it will stain dark. Euchromatone stains much lighter. It's more transcriptionally active. The transcription factors enhances all of that can get to the sites more appropriately, and it is less dense. The reason why it's helpful to have all these numbers and names for each of these segments is it's useful in identifying abnormalities and breakpoints, as well as describing the location of particular genes. Next slide, please. These are the types of mutations that exist. A silent mutation produces no change in the amino acid and therefore no change in the functional protein or its conformation. These will often occur in intron, which, if you remember, on non-coding regions of the DNA, or it will occur in a wobble-based codon. So if you remember when the mRNA is divided in triplets, and the third one is susceptible to wobble. So there's redundancy. So if that mutates, the same amino acid is produced and the same polypeptide is produced. It is not changed. There's something called a missense. So I did the French accent that time. And that is a change in one nucleotide. And this change in a nucleotide will change an amino acid produced and may result in impaired function of the protein or it may enhance the function or reduce the function of the protein. A nonsense is when there's a premature stop codon that terminates the synthesis of the polypeptide. I'll fix that up before we publish these slides. Of the polypeptide, which means that there's some mutations that happens that made the triplet codon red the stop codon that's going to bind a release factor while the mRNA is bound to the ribosome, and you're going to have an incomplete protein conformation, and that protein is not going to be functional, essentially. There's also something called frame shift mutations, which is when a nucleotide is added or taken away from the sequence. So if you think you have 3 things here, let's do 4 here. Usually you read the first 3 in triplets, but what's happened is you've had a mutation that's taken this away, but then these 2 go together. So then instead of these three being the original triplet, these 3 become the triplet. And then these 3 become the next triplet instead of initially being these 3. I hope that makes sense despite my messes, but everything shifts, everything changes, all the amino acids change. It completely alters the protein if it doesn't produce a premature stop coding and lead to a nonsense coding first. Tell me if that doesn't make sense. So next one.
Yeah, so silent and missense aren't as deadly, but nonsense and frame shift will majorly screw you over.
Exactly. So breakages or rearrangements can be balanced or unbalanced. and they may result in the spontaneous miscarriage due to a non-viability of a developing embryo. The embryo needs certain genetic material, certain proteins to make, certain enzymes to survive. Or the child may be born and carry abnormalities if the embryo is viable. So usually a balanced rearrangement will lead to a viable embryo. Balance means there's no genetic material gained or lost, it just changes position. So it's still going to be transcribed. Unbalanced means that if genetic material can be lost, which means really important proteins may not be able to be transcribed and may lead to the non-viability of the embryo, or it could be a gain of genetic material that is also unsuitable for life. These abnormalities may occur both to autosomes or sex chromosomes and may affect the structure or number of chromosomes. These are nice ways to classify it in your mind. We'll go through this later, but remember structure or number. You can have more chromosomes, less chromosomes, or the chromosomes can shift around. Next slide, please. Numerical abnormalities. Aneuploidy means there's an abnormal amount of chromosomes in the cell. That's all, it's very general. Monosomy means one member of a homologous chromosome have been lost. Trisomy means there's gain of a single chromosome, such as in Down syndrome, Edwards syndrome, and Patau syndrome. You need to know all of these. We'll go over them in a bit. And you have tetrasomy, which is gain of 2 chromosomes, low yield and quite deadly. Polyploidy is a multiple of haploid number in cells. Most of these end in miscarriage. So essentially it means if you usually have 46 chromosomes, during the formation of the zygote, when you have a sperm or an egg, you might have an additional gamete being part of the zygote. You're then going to have a whole 23 more chromosomes, and it goes up in integer layers in a polyploidy. These are very dangerous, and they're only relevant in megakaryocytes, which form platelets, which is low yield, and you don't need to know. Next one. Most aneuploidy results from non-disjunction of the sister chromatids during anaphase. As you remember, anaphase is when it's breaking apart. This means they haven't been breaking apart. And it can occur in either meiosis 1 or meiosis 2. If aneuploidy occurs during mitosis, then there'll be 2 cell lineages with different karyotypes. And this condition is called mosaicism. So let's say this is your zygote. You have these cells proliferate. You have a non-disjunction in mitosis here. So this cell is mutated. This cell is not. This cell is going to go on and it's going to be normal. And this one is going to have the mutation. So there's some cats that you can see their different colors are due to this mosaicism, different expressions of genetic material. Yeah. That's why it's worse if you have a mutation in the zygote or during the meiosis stages that form the gamete than if you have it during mitosis. Next one. I'm going to try and speed it up a bit because we've got a lot to go through.
Yeah, I think the diagrams at the bottom are pretty useful to visualize it though.
Yeah, exactly. Again, this is the mitosis. If you want a more visual understanding, you can look at these. Next one. Important syndromes to know. Edwards syndrome is an additional 18 chromosomes. This is highly examinable, will be on your exam. Patau syndrome is an additional 13 chromosome. Down syndrome is an additional 21 chromosome. That's exams probably not going to be more than that. You'll have an ICL where you're going to have to go into different types of Down syndromes. Most of them are because of a complete trisomy. 1% are because of the mosaicism in the mitosis we were talking about before. And 4% are because of unbalanced rearrangements and Robertsonian translocations we'll talk about later. Know what's in red. You know what's in red, you'll get the marks on the exam.
Yeah, you don't need to know the specific numbers, but just know what the actual disease is.
Yeah, exactly. Klinefelters are clear. Klinefelters, XXY, often leads to infertility. Turner syndrome is just X0, so 1X. Know these. Very high yield. The rest you can read to understand what you're doing, but if you just want to get the marks on the exam, just know those. Most polyploidy, so this is very low yield, most end in miscarriage. You can see how there's been an extra gamete that is fused in the zygote. 3, 3, 3, 3. You can see these are the different types of possibilities that occur, which is just variations of the gametes. And polyploidy occurs in megakaryocytes, which isn't important. Next slide. Structural abnormalities. These are the types of rearrangements. They often can break and rejoin. So translocations, insertions, duplications, inversions. Let's go through them. Next slide. Again, we said balance is no let loss or gain. It includes inversion, translocation, insertion. But however, the carriers are at risk of an unbalanced rearrangement, which is important in Down syndrome. Or unbalanced is net loss or gain. Okay, unbalanced, clinically severe, net loss or gain. Balanced, no let loss or gain, usually harmless, may be harmful in the offspring. Okay. Breakage of the chromosomes. Sometimes chromosomes do break apart, and this piece may either be permanently lost, it may be duplicated and then reincorporated, and it may break off and then be reincorporated in the wrong direction. These are, oh, go back, sorry. Deletion, you can see there's a loss. Duplication, you can see it's addition. Inversion, you can see it's switched. Next one. We're almost done. There's 2 more slides, I think. Genetic material may break off and it may be reincorporated into a different chromosome. So chromosomes can swap. This is called translocation. It can also just insert without the reciprocal nature. Know these words, know what they look like. If you just remember the pictures, that's good enough. Next slide. Because if you look at the pictures, you'll know what's balanced, what's unbalanced. Reciprocal translocations is when part of the chromosome breaks up and these fragile attach to the origin of each other. So you can, here is an image to help with this understanding. You can see these have translocated. And you have a partial trisomy as a result of the additional genetic material of these, and a partial monosomy because these are not complete chromosomes. Important in Down syndrome. Again, you just really need to know the word. Do people agree with that, or do they go over the concept and make sure you understand this? But I think if you just know translocation means they swap genetic material, can lead to partial trisomy or partial monosomy because of the loss and gain over here, then you'll be all right. Next slide. Robertsonian translocation is an example of a clinical implementation of this translocation. I cut off some of the words there. So the satellites that we've talked about in acrocentric are lost. And that means the long Q arms combine together and fuse at the centromeres. So you can see that here. This bit and this bit were lost for this chromosome. And the Q-tips, the long tips fused together. You can see the consequences of this is you can have Down syndrome, for example, where in mitosis, these two are brought to one cell. This is brought to the other cell. And then when they're fertilized with another zygote, which has a 21 and a lon head, so the 21, this is the other zygote it fertilized with, you're going to have 1, 2, 3 21s. You have trisomy 21, which is Down syndrome. Hope that makes sense. Really just 10 minutes, go through the drawing, you'll get it forever.
Yeah, so yeah, the drawings are really helpful. Sorry going a bit quick, but we don't want to keep you guys here for too long because we know you have other stuff to do.
And we also have lipid metabolism, which is huge next, and other stuff. Insertional translocation is when insertion of a chromosome from one chromosome to a non-homologous chromosome. Nothing is lost, it's just moved there. Next slide. Okay, lipid metabolism. Okay, now with this one, I made this as comprehensive as possible. So we're not going to go through everything that isn't important, but I'm going to make sure you have a good idea. And I understand that you might not be as exposed to this as, so we'll go nice and we'll go slow, be very clear. You know what a triacylglycerol is? It's 3 fatty acid chains attached to glycerol. They are hydrophobic, non-polar in water as a whole, but there are hydrophilic, which is water-loving ends, and there are hydrophobic, which is the non-lipid ends. They're used for energy storage and diets. The rest, do you need to know? Yep, phospholipids constitute the cell membrane, 2 fatty acid chains or phosphate group, and sterols are used. More important later, steroids you need to know, important in maintaining cell membrane fluidity, but sterols can also be used in paracrine hormone signaling and gene expression and lots of things which you don't need to know currently, but you will need to know soon. Next slide. Fatty acids, 3 types. Everyone's done chem, so you might remember. Saturated, they're only carbon-carbon single bonds. Monounsaturated, which is there's 1 carbon-carbon double bond. Polyunsaturated, clinically significant, 2 or more carbon double bonds. Essential fatty acids are polyunsaturated fatty acids, cannot be synthesized and must be consumed in the diet. Next slide. Essential fatty acids must be required from diet and cannot be synthesized. 2 huge types required for physiological, omega-3, omega-6, low yield for interest. These are important. Polyunsaturated fatty acids are really important. They constitute the cell membrane. They're involved in hormone signaling, cell signaling, the inflammatory process, clot formation, smooth muscle, lots and lots and lots of stuff. This is not high yield. All you need to know, polyunsaturated fatty acids cannot be synthesized by the body. Omega-3, omega-6, max. Next one. And you should maybe know they're important in cell membranes. Omega-6 is used by the body to synthesize ixanoids and in the form of arachidonic acid. That's what it's used to synthesize. So you can see linoleic goes to arachidonic. I can't pronounce it, sorry, but this is hugely important in the formation of lots of signaling and stuff. Not important. Next slide.
Arachidonic acid might come back later in pharmacology when you do it.
Yeah. Like these are important concepts, but they're not important concepts for lipid metabolism right now. Yep, we said that. Paracrine hormone means it acts on nearby targets. Yep, we said all this. Next slide. This is just so you trust me. Prostaglandins are involved to elevate body temperature in pain and inflammation. Thromboxane is involved in blood clotting. Aspirin acts on thromboxane to inhibit blood clotting, which is why aspirin is a blood thinner. Yep, next one. Low yield. I'll tell you when we're high yield. The low yield, skip it. You can look at that if you're interested. Very high yield. Fatty acid synthesis. You learned about beta oxidation last week. This week, we are not just doing the reverse of fatty acid oxidation. We're using different enzymes in different pathways in different locations. The major sites of fatty acid synthesis is the liver. However, it does occur in adipocytes during glycolysis as well, which you should know about, and it does occur in muscles and mammary glands, not as important as liver and adipocytes. It occurs in times of plenty. You have high carbohydrates and low fats, fatty acid synthesis will be promoted because, as you know, triacylglycerols store a higher concentration of energy. If there's low carbohydrates and high fats, fatty acid synthesis is reduced. There is a negative feedback. Next slide. Sorry, I'm going quickly. If you don't stop me if you don't understand, but we've got lots to do. Conversion to melanonucleoid is the committing step. You cannot go back after that. This is a high yield concept. After melanonucleoid, you combine with NADPH. This is your introduction to NADPH. It is also it's very similar family to NADH and FADH2, similar function. It produces fatty acids. Next slide. You went through something similar last week. They like to use 16 carbons as an example. This is a formula to know what are your ingredients required to synthesize fatty acids. Something you can probably look through in your own time. Important to remember, you require ATP. Yeah. I'm very, very unlikely to ask you to do this in an exam, but it is just maybe just x divided by 2, x divided by 2 minus 1, x minus 2. You can think about it as well as you're synthesizing. You need 2 acetyl-CoAs to make the chain. Yeah, it's okay. This is something you can just remember it if you like. Next one.
Yeah, I think this slide is too important.
Agreed. Just ingredients. Synthesis in the liver. For some reason, this was reasonably high yield. When we're in fat cells, in order to synthesize fatty acids, you need to be in glycolysis. That doesn't make any sense. Glycolysis is a breakdown. Fatty acid synthesis is when you synthesizing energy. Why does that work? The reason why that works is from glycolysis, you get a product called glycerol 3-phosphate, and that combines with the fatty acids to make triacylglycerol. Important to remember, fatty acid synthesis in adipose tissue only occurs during glycolysis. It's been in exams, know that. In the liver, there's a bit of a different pathway. It's you have glycerol that glycerol kinase acts on glycerol, makes glycerol 3-phosphate, and then again you're adding the fatty acids to this glycerol 3-phosphate. I think you can picture this. It's very chemistry. You can see how this looks like. You're just going to combine 3 fatty acids to this, and you're going to get triacylglycerol. Next slide, please. Hope that makes sense. That's quite important. Cholesterol assists in cell membrane fluidity and is part of the cell membrane structure. It's a precursor for steroid hormones and biles. And okay, what you need to know, associated with cell membrane fluidity, associated to LDL, which is very highly associated with cardiovascular disease. And we'll come back to it. LDL is also known as bad cholesterol. This is you're getting real medicine now. Let's go. Next slide. Pathway, statins act here. That's why they tell you you don't need to know about statins yet. Actually, you do. They do ask. Statins act at this enzyme called HMA-CoA reductase, which is a limiting enzyme and limits the production of cholesterol, which means you're going to get less production of bad cholesterol, which clogs your arteries, and we'll see why. Next one.
Yeah, this enzyme is very important.
HMG-CoA reductase. Comes up over and over again. Lipoproteins, they're these little structures. Why do they exist? They exist because usually triacylglycerides or fatty acids are nonpolar, and so they're not soluble in blood or plasma. When they're surrounded by these little phospholipid heads, they're polar and they're soluble. They have a hydrophobic core with tags and cholesterol esters. Why am I drawing over the words? And hydrophilic shells with phospholipids, cholesterols, and apolipoproteins. An apolipoprotein plus a lipid equals lipoprotein. Apolipoprotein is just a protein that protrudes from here. Next slide. It's a signaling protein. High yield, know this. 4 big types of lipoproteins to know. When you eat your food, it's going to eventually, the fatty acids will diffuse across the enterocytes, which are in your small intestine. We'll package them into chylomicrons, which are mostly tags, 80% tags. And these will drain into the lymphatics because they're too large to go into the blood vessels. In the lymphatics, they'll be broken down by lipases, which will release glycerol and fatty acids to the appropriate tissue that requires it. As this is occurring, you're losing fatty acids. And once you get to around 56% tags, you have something called a VLDL, a very low density lipoprotein. These can be transported. No, sorry, not yet. The chylomicron is going to be broken down by lipase. It's going to reduce in tags, and something called a chylomicron remnant will return to the liver. In the liver, VLDL will be synthesized. I hope I didn't confuse you. Chylomicron goes around lymphatics, goes into blood vessels, broken down by lipase. Glycerol fatty acids go to tissue. Chylomicron remnant goes to liver. Liver synthesizes VLDL, and this is transported from liver to tissues to supply them with tags. It will return to the liver as itself, or it will become LDL. What is LDL? LDL is a low-density lipoprotein that deposits cholesterol. It is mainly cholesterol. It penetrates vascular endothelium due to its small size and is involved in atherosclerosis or path buildup. Often, HDL is then synthesized in the liver as well, and it is good cholesterol, whereas LDL is bad cholesterol. And good cholesterol esterifies the bad cholesterol, picks it up uptakes it, returns it to the liver to be recycled as bile, etc. Next slide is going to scare you. Next slide.
I just quickly might add something. So you
Yeah,
can see the change in density. So you can see HDL is higher in protein, which is more dense. And as you lose more tags, you become more and more dense, I guess.
Exactly. Next slide. Scary. These are for your notes, essentially. I put them, they go up the whole process. This image is beautiful. You should recognize it. You've swallowed your food, enterocyte is packaged as a chylomicron. Chylomicron is empty the lymphatics. It's been broken down by lipases. Wrong direction. Hold on. Undo. Where are the lipases? Okay, lipases are in here. It's being broken down into lipases. Free fatty acids are released to appropriate tissue. Remnant goes to the liver. C for cholesterol, C for chylomicron. It's recycled. You can see VLDL is made. The VLDL becomes IDL as it releases free fatty acids. IDL becomes LDL. LDL becomes foam cells. It doesn't. LDL deposits on the endothelium cell wall. Then macrophages in your cell wall will phagotize them. Eventually, the macrophages are saturated with them and they become foam cells. They start to protrude in the endothelium, in the tunica intima of the blood vessel, and plaque buildup occurs. This will narrow vessel walls. This is what angina is. Real medicine now. Or it may occlude the vessel wall completely, or rupture, occluding the vessel wall. If it ruptures, everything in that plaque releases out, and that is called a heart attack or a myocardial infarction. I highly recommend you go through this in your own time as your notes. Next slide.
If you manage to understand the diagram, you'll be set. So spend some time here. We are going through it a bit fast, and you don't really need to know about angina and MI right now.
Yeah, you absolutely don't. I just did that because I thought it'd make it more interesting. Some LDL does re-enter the liver through receptor-mediated endocytosis, and there are certain conditions when the receptors that allow LDL to come back are defective, and these people have a greater chance of having cholesterol buildup in their arteries and are more susceptible to cardiovascular disease. Not important, but just so you know, this is why they teach this to you. It's important. Recycled cholesterol is used to make HDL in the liver. It has mostly protein with very little cholesterol. It goes to the epithelium, estrophises cholesterol, and takes it back. Next one. Almost done.
I might also mention that we talked about IDL, but like in the diagram, we didn't actually go through it. IDL is just between VLDL and LDL, but IDLs don't get mentioned very much for some reason.
No, it's because they're just intermediate. Like they're kind of neither here nor there. VLDL mostly tags, LDL mostly protein, IDL is in between. It's not really important. It distributes free fatty acids broken down by lipases. Next one. You should know this. Very high. Oh, it's not very. Yeah, very high yield. Know the picture. Apolipoprotein is a receptor protein on the column on the LDL particle complex. The LDL particle is going to interact with these receptors and is going to be uptaken by an endosome through endocytosis as the LDL is internalized. What's then going to happen is there's a recycling mechanism in which the receptors are returned to the surface. Well, the LDL is taken down, it fuses with a lysosome, creating an endolysosome, and lytic enzymes are going to degrade the cholesterol-free fatty acids, amino, everything to be recycled so that the liver can synthesize VLDLs, bile. You mentioned it, liver makes all of this. Know this picture. Know the receptors are important for pathology later on. That's why they teach it too. Next one. Atherosclerosis. This is a greater explanation of it. These steps make it easier to think about as well. You're going to have endothelial injury. That's going to make the endothelial wall susceptible to infiltration of LDLs. Then macrophages in the tissue. Oh, what's the word? You don't need to know what the word is, but macrophages in the tissue will then phagocytose the LDLs and become saturated and become foam cells. Platelets in the blood that circulate and form clots are going to start to adhere to the damaged endothelium, Because usually when you damage your skin, it clots so you don't bleed out. So it's going to clot as well. And that's going to build this mass filled with foam cells, clots, and clotting factors. That's going to build and build and build. Your body is going to treat it as if it's a vessel wall and is going to make smooth muscle to keep its structure. It's going to have a fibrous cap. It's going to calcify to make it stronger. If at any point this cap bursts, all this contact is going to rush out, and that can cause an MI. As this constricts, you're going to get reduced blood flow. Don't really need to know that too much. You just need to know the mechanism of action, as well as what happens is the HCLs will then come to these sites and estrify the cholesterol, making them taking them back into the liver where they can be recycled. Next slide. Another picture, if this helps you think a bit better. We've been through it. Next slide.
We'll come to stick around after as well if you'd like to ask some questions.
Yeah, okay. So time for the best subject, HKS. All right, so we're going to go through health determinants today. And basically, this is kind of like what HKS is, kind of. Describes how different factors affect the health outcomes. And today we'll be going through health determinants. So just some definitions you kind of should know. Factors and determinants, they're just basically used interchangeably in HKS, so don't worry too much about it. Social factors / determinants, these are factors that are basically inherent or existing in the social environment. And then there's structural factors / determinants. And there are all of the social, cultural factors and factors which may occur geopolitically, legally, historically, environmentally, and things like that. And they basically affect the ability to make our decisions. So as you can see on the right, this is basically how we categorize them. So basically social factors are part of structural factors. Next slide, please. All right, so this is a really good analogy that you will be taught. It's called the river analogy. So you have an upstream and a downstream in the river. And that's kind of like the same in medicine as well. So in upstream, you have different social and structural factors impacting someone's health, maybe even without this person ever, going to the hospital, right? And downstream is the outcome of that, so the disease and the health outcome. So if, for example, on top of the river, if someone puts down poison down the river, then the downstream or the things all the way down there, it's going to be all poisoned, despite the fact that there was nothing wrong with that in the first place, even though it was just upstream. But that's like the flow of medicine as well in HKS. So if you go to the next slide, because of this, we want to try have upstream interventions and downstream interventions as a health professional. And downstream interventions are what basically we all think about when we talk about medicine, the treatment of disease, right? So if someone comes in with a cold, you give them the treatment that they need for the cold, right? That sounds pretty simple. But medicine isn't just evolved around just treating the symptoms when they come in. And we want to kind of address the issues, if there is any, that may have caused this health outcome. And that is the upstream interventions. So, maybe this person was a refugee and they were displaced from their homes. So that was going to have a great health outcome or impact on the health outcome. And, we should also be thinking about those different things that are affecting the health outcome and not just the disease themselves itself when they come in. Also, things like vaccination and preventive medicine are really good ways to try to prevent the social determinants of health or structural determinants of health affecting it. Next slide, please. So these are some of it, not all, but some of the social determinants of health. So again, as I said, they're inherent factors that affect health of individuals / communities, and it could be positive or negative. Like for example, job status. If you have a job, it's kind of reported that you're more happy. If you don't have a job, you're less happy, right? So it can be positive or negative, but in the end, it's still something that will affect you. And also there's this thing called social gradient that one of the PSPs last year talked about, not necessarily in our lecture, so I'm not quite sure how relevant it is. But basically, it's a thing saying how more wealth equals greater likelihood of good health, sorry. Because you have more money to basically not only get the treatment, but also live in a scenario and an environment where you are happy and your well-being is great, right? So other things are like geographic isolation, gender, discrimination, addiction, all these things will affect your health negatively or positively. So next slide. So that's social. Now, structural factors are basically on a bigger level. So these include four main things that you'll be talking about in HKS. The first one is geopolitics. And this is just, as the name kind of suggests, like to do with geography and / politics. So like kind of like your whole countries in the conflict, maybe, you're maybe displaced because of a war, or like neoliberal globalization, which you will also talk about. And social cultural factors are like social determinants, cultural blindness, and cultural safety. Now, this is something that you'll be going through your Aboriginal modules, I believe, Aboriginal health modules. And they basically, you can be inherently, or you cannot be inherently, you can be discriminately racist. Like you could be like saying slurs, things like that, and being racist. But also you could kind of be racist without actually acknowledging or even knowing the differences of culture. And that's kind of cultural blindness, not really recognizing that there's differences in cultures, and to basically to respect that. Even though you didn't actually do anything, the fact that you didn't do anything in a way could be racist, or not racist, but could definitely be cultural blindness. There's also environmental change, so like climate change, which is something that's really big nowadays, urbanization, all of these affect your health outcomes. And history and legal factors, especially for Aboriginals and Torres Strait Islanders, they have colonizations, the legislation, health policies, all of this in the past, but also in the legal system that's affecting the health system and the health outcomes of some of these people. That's really important to know because you will come across some of these words. Next slide. There's mediators. This is pretty straightforward. So just like what kind of can you do to change the health outcomes? So like changing basic needs, behaviors, access to healthcare, which we'll go through more about in the next lecture, exposure to hazards, changes in psychological well-being, but also direct physiological changes. Next slide. Structural violence. So structural violence is something that came up in like the late 1990s, and it's basically a biosocial-political framework. And the structural in the structural violence refers to how it occurs within the local, national, or international social political framework. So it's something that kind of isn't in your control in a way, kind of because of the government or because of a war next to you, or because of the war that your community started. And then there's obviously violence because it causes an injury, directly or indirectly. So basically what structural violence is it describes how asymmetrical structures of the knowledge of power / politics can interact with the social structures to create the health inequities. So that's a good example would be refugees or people from the war zones. So that's all for HKS this week. It's really important for this week is kind of just knowing that there are different factors affecting the health outcome, and we should kind of do something about it, not just treating the disease, but also the factors that may have affected, in a way, the upstream of their life. So next slide.
Yeah, so I think an important point is that with the upstream, downstream things, you want to target the upstream things. So, like you could treat them when they're sick, but ideally you'd want them to never have gotten sick in the first place. And that's the main point with this upstream, downstream thing. The factors and stuff, just know them generally. They're sort of intuitive as well because, if they have a poor education or, they live in a war zone, that's probably not going to help them well-being. So it's fairly intuitive, but just with HKS, it's really important that you just get the general big ideas and the big picture that they're trying to get across.
This week, and you will have an assignment for it as well. So in the exam itself, ethics isn't really examinable, not examinable, like there's not many questions about it, but there is an assignment for it, so you need to know about it, even though it's a pass or fail, but yeah. Next slide. So what is ethics? So it's the moral principles that govern a person's behavior or conducting of an activity. So because it's kind of really subjective, over the years there have been different people coming up with different ideas of what they think about ethics really is. And one way to approach ethics or consider ethics is to think about the extrinsic and intrinsic values. These differ from person to person. So what extrinsic values are like? Values which is, the value is determined by what we can do with it. For example, knives are useful because they're useful for cutting. mobile phones are useful because they can be used for communication, things like that, right? And PSP Peppa Pig is useful for passing exams, hopefully. But intrinsically, you can also have the values think about it as intrinsically, and that's like, the value of itself for its own sake, right? Like happiness for happiness, kind of, right? It's kind of like something that's intrinsic, doesn't really have, the value isn't determined by what we can do, but rather just what it is. So because of this, it kind of sounds all good, but then there's this thing about a conflicting example about lying. So lying is bad because you might hurt someone with someone's feelings, right? Extrinsically, that's an extrinsic approach. But then you could also say lying is bad because you should never lie, intrinsically, right? So is, lying extrinsic or intrinsic, right? It's kind of hard. It might depend on the situation. So we have white lies as well. Like it might be better to lie, but then intrinsically, if you might believe lying is bad, then you never do white lies. Where you might do white lies because you think it won't hurt them, rather it will help them, right? So extrinsically. So this is kind of a little bit of a conflicting example, and there's a lot of that. So you will need to kind of have your own mind over what you think about certain situations. And for the assignment, depending on the situation, you'll need to apply the ethical theories and the 4 pillars of principles that we will talk about in a short time. So next slide, please. So there's ethical theories here. These are just kind of mentioned in one of the lectures, but principle-based is kind of more important in my opinion. So don't need to worry too much about the exact, theories of this, like who made it, why is it, but you would need to know roughly what they are. So we'll go to the next slide. So I can't say this word, consequentialism, okay, or utilitarian, I can't say words here, sorry. But basically what this is, it's a theory saying we can say an outcome is, or we can say something is right or wrong depending on the outcome, right? So this sounds all good in theory, but the problems kind of come across if we go through some examples. And one of the examples might be like, you might be okay with one person dying if it's to save 6 people. But what if that one person dying is a close member of the family, right? Sorry. Then that kind of gives a conflict within yourself. So utility is like really like kind of like all like paradise kind of thing, where it's like really good if you can do it, but in theory it's really hard. Now there are some other words that are some other sentences you can read later, but not important today. So we'll go through the next slide. Okay. Now deontological ethics. So this is talking about how it's not about it's not about wait, sorry, let me get some water. Sorry.
Do you want me to cover?
Oh, I think Peter's dead.
Yeah,
So
he's gone.
yeah, mainly of
sorry, yeah.
deontological stuff. Well, you can cover with me, David, because I don't know if I remember everything. So
Yes.
deontological is basically you act by your duties. Like you have these rules that you should never disobey. So there's something like there's like a Kantian who was like a philosopher, and he had this little thing where it was like, oh, lying is bad, so you should never lie. And he was like, even if a murderer is asking where your friend is hiding, you shouldn't lie because that was a rule you set. So even if your friend dies, you shouldn't lie anyways, which is a very extreme example. But
Yeah.
it's basically what deontology is. It's that consequences aren't really the important thing here. What's important is that you follow what you set out beforehand and you adhere to it.
Yeah. So just to summarize, deontological means actions are either inherently moral or immoral. You never lie, you never steal, regardless of the circumstances. Consequentialism is you weigh up the consequences of your actions to decide whether it's the right or wrong thing to do.
Yeah. Okay, maybe the next one then. But I might add, like, with this consequentialism at the bottom, obviously, if you strictly adhere to it, then you'd have to give away all your resources to help others because, there's always people in worse shape. But if you do that, then obviously you lose everything. So I think there are like some people have said like you aren't like extreme consequentialism isn't something that's really viable. Communitarianism, I believe that was basically like helping out the community, right, David?
Yeah, very low yield. There's a sentence of it in a reading.
Yeah, it's not very important.
But it essentially means all individuals, our society is underpinned by the fact we all exist in a wider community, and therefore we should do what we can to support the community.
Yeah,
For
it's just like a
example,
community mindset.
yeah, so and that can sometimes inflict on individual liberty. So you can't, vaccinations are compulsory, even if you don't want to, for your kid to go to childcare. Yeah. This is the best one. Virtue ethics is from Aristotle. It's the concept from him. And it's the idea that trying to define general actions of general philosophies of consequences are better, or on what you're doing, or certain actions are right or wrong, is misguided in the fact that it kind of takes away the human element to it. Virtue ethics is saying that to do the right thing is to be a good person and to do what a good person would do. Be
So-
like a good person should be, rather than think about what to do. And when you are like a good person, then you will act like a good person.
Yeah, so it's basically you have these ideas or principles that you should exercise justice. You should be selfless. There's no specific like rules like, oh, you should never lie. But really, depending on the situation, do you lie or do you not lie? Just like depends on which would be the right thing to do, or which would be more virtuous according to what your virtues are. It's just kind of like following some principles you set out, I guess.
And the way to know that is you make yourself the best person possible, and then you trust your judgment.
Yes, just be a good person, David. Why can't you do that?
Dare you. Next one. Getting called out. The most important part of these slides, because these will be in your assignment. 4 principles for healthcare, drawing consequentialist and deontological. These, okay, you don't need to know the history of it, but Andrew, would you like to go through it? Or?
Yeah, I can try. So this is what's most important because this is what we use in medicine. So yeah, as we said, it's a bit of consequentialism where you care about what happens to the patient, obviously, but it's also deontological where you have some rules that you shouldn't break, such as privacy for the patient, that kind of stuff. So there's 4 main pillars. So there's respect for autonomy, there's beneficence, there's non-maleficence, there's justice. It's fairly simple to go through. So respect for autonomy is just really recognizing the patient's right to do with themselves what they want to do. They don't have to get treatment. They don't have to, do what you say. You have to respect what they want for themselves. And so an important part of that is getting informed consent from them. So you either have to, they have to be capable of giving consent. You have to give them the required knowledge for them to give consent, because you can't just tell them nothing and tell them to say yes. You need to give them the information about maybe a procedure, stuff like that. And you cannot coerce them into giving consent. So it has to be free consent given by them voluntarily with enough knowledge provided. Beneficence is basically saying that you should really try to do good, do as much good as you can, and minimize risks of harms.
Beneficence is about weighing things up. So what is going to have the best outcomes with the least risks? Beneficence is a pro and con list, whereas non-maleficence is saying do not do harm, which is like don't show up drunk, don't hurt people without evidence. So lots of people get confused between the difference between beneficence, which means to do good, and non-maleficence, which means to do no harm. I think for beneficence is the weighing up. Non-maleficence is like don't do something really bad.
Yeah, there is a point that there is non-avoidable harm. Like if you have to, stick a needle in a patient, that's harm still, but that's unavoidable harm. So non-maleficence refers to do no avoidable harm. So that's a key difference. And then justice just refers to you should treat all patients equally. Resources should be fairly distributed. You shouldn't discriminate, stuff like that. So just be a decent person, basically. And I think that's it for principle-based. there much else? I think this might be the end, right, David?
Yeah, I think so. And also justice about equitable distribution of resources, like you said. One of my friends forced me to watch a Grey's Anatomy episode last night. And there were these two people in really bad conditions, and they had to choose, do they let the one die to save the other one? That's justice. That could be in your assignment. You have to say who deserves treatment the most. I reckon what they'll do with you guys is, you know how in Italy they had to choose who got treatment and who not because they all had COVID. I think they'll give you a situation like that, and you'll have to be, well, do the most vulnerable people need the treatment, or the people who are most likely to get better need the treatment? And you'll apply all these principles to do that.
I think what Italy did was like they try to treat the young people first, because they took a consequentialist approach where if they treated the young or like more likely to survive patients first, they'd probably save more people.
Yeah. So keep that in the back of your mind. I think we're done. We've kept you for a really